Okay. Well, great. I am delighted to welcome everybody back. My name is Tommy Source. I'm the Assistant Director of the Purdue Institute of Inflammation, Immunology, and Infectious Disease. And I am excited to welcome you to another Research Spotlight series talk. Today, we are honored to have Dr. Arun Bhunia, Professor of Food Science and Food Microbiology in the Department of Food Science. He's been dedicating his life really to understanding and trying to affect ways by which we can counteract infections from pathogens like Listeria. And his talk today on host cell receptor targeted bioengineered probiotics for prevention of infectious diseases is spot on to our group here at PI4D. And welcome, Arun. Thank you so much for doing this talk for us. Welcome, everybody, and thank you for taking time out of your day to join us again. Without further ado, I will pass the microphone back onto you, Arun, to take it away and tell us about your great work.
Thank you, Tommy. You can hear me, right?
Yes, loud and
Okay.
clear.
Okay, excellent. Good morning, everyone. I'd like to share some of our work in this area. Like Tommy said, our research focus primarily is Listeria monocytogenes. So definitely I'm going to talk a little bit more about Listeria before I get into the receptor approach we took to control this infection. Um, So I'm going to use actually 4 research articles as a model to give you the highlights of some of the work we are doing. And hopefully it's not going to be too long. Try to stay within the time window. I know many of you have other activities, especially this is the final week. So let's get to this. So as I said, Listeria monocytogenes is the primary pathogen we are focused on. This is one of those model pathogens. It is an intracellular pathogen. And as you can see in this slide, the Listeria in a Keiko2 cell model interact with the cells and then invade the epithelial cells. And clearly it shows a nice way of Listeria to enter into the cell. So it's a gram-positive pathogen. Let me give you a little bit background about the importance of Listeria as a food-borne pathogen. First, it was isolated way back in 1926. It was in Cambridge, and there was an outbreak of Listeria in rabbits and guinea pig. And so that's when first this organism was isolated. And then it's primarily known as a animal pathogen, especially ruminants. Cattle and sheep are primarily infected. And when they get infected by Listeria monocytogenes, especially it infects the brain, so you see the circling movement, animal move in circles. And so you see signs of meningitis, encephalitis, and septicemia. And also occasionally it causes abortion in animals. Um, as a pathogen, as a foodborne pathogen, US, especially we reported early in 80s, that's when first large outbreaks of Listeria was recorded. And ever since, we keep seeing a lot of outbreaks of Listeria with the food. So one of the pathogens, so that's the Listeria monocytogenes, that's the main concern in humans. And Listeria ivanovii primarily infects animals. We have seen cases of Listeria ivanovii infection humans as well. Non-pathogens, just one organism I want to highlight, Listeria inaqua. It's look like Listeria monocytogenes, but it doesn't have those virulence genes. So it's a non-pathogenic. People use that as a surrogate organism for some of the studies. Also, there are 17 more non-pathogenic Listeria, and recently people identified several of them. As a foodborne pathogen, gastrointestinal environment is very critical for Listeria. And so Listeria has found a way to survive in that. Just to give you one example, like glutamate decarboxylates, that's helped them to survive in the low pH conditions. Also, it has defense against bile. So this number of ways they can survive well in the gastrointestinal tract. And in terms of number of cases, we see about 23,000 cases annually. That's in the global report. And in US, we see about 1,600 cases and 260 deaths per year. But this is the foodborne pathogen that has the highest hospitalization rate. So obviously, you can expect a large, cost associated with this infection. And as a foodborne pathogen, obviously, it has an interesting lifestyle. So we call it one saprophytic lifestyle, and other is the host. So obviously, they need to survive and survive well in the environment, especially soil, decaying vegetation, sewage, and silage. That's the main feed for animals, and that could be a major source of listeria. So animal feed also being implicated as a source of Listeria as well. And most importantly, human foods. So you can see the list of foods being reported as a source of this pathogen. Uh, Soft cheese is one of them, was responsible for several outbreaks, especially the queso fresco. Ice cream also. I think recently just one recall being or outbreak, I think, with the ice cream right now in progress. Cantaloupe, that's one of those happened in 2011. Several people were infected and also dead. And polony, this is another interesting food product of South Africa, actually, called French polony. It's almost equal to like a bologna here. And it was a source of Listeria, one of the largest outbreak, about 1,000 cases, more than 1,000 cases, and over 16 people died, actually, 2017, 2018 outbreak. So that's the reason, I guess, US and Brazil has a zero tolerance. We don't allow any Listeria in food, especially Listeria monocytogenes. And Canada and Europe, they allow certain numbers, about less than 100 colony forming unit. And they have different directives, like make sure the bacteria do not grow in that product until their shelf life. Anyway, some other countries allows that. But if you look at the infectious dose, it's pretty low, actually. As low as 100 cells, or even lower, and also as high as 20 to the power 11. So that's kind of broad range. It primarily infects immune compromised people. Healthy adults are generally, we are of low risk, but we may suffer from febrile gastroenteritis. The mechanism of gastroenteritis is not really known, so we may need to find out a little bit more about how this organism causes gastroenteritis. But primarily, it causes systemic disease. Pregnant women, the fetuses, the newborn, they are heavily infected, and it's pretty severe infection, you'll find. So overall mortality rate is 20 to 30%, it's pretty high. So in terms of listeriosis, so as I said, gastrointestinal listeriosis, it causes rare diarrhea. There are about 4 or 5 outbreaks they have reported. But again, the mechanism is not really well understood. The systemic listeriosis means once the bacteria is ingested, and then it spreads to different parts of the body, and that's well understood, how actually organisms survive in some of the organs and tissues. And then also the abortion or neonatal listeriosis. So in terms of understanding the disease, so Listeria has to pass through the gastrointestinal barrier unless it's causing gastroenteritis. But mostly it has to pass through the GI barrier, and then you'll find them in the lymph node in large numbers. Then eventually you'll see in the blood circulation. Liver is one of the primary sites you'll find large, high loads of Listeria. Then also in the spleen. And if the infection persists, then you'll eventually find them in the brain, and it can cross the feto-placental barrier, then can infect the fetus. So in pregnant women, that's a major problem. So one of the questions we try to ask is that, how do Listeria cross the barrier? And so obviously there are some studies have done, and I think it's not fully understood how the how Listeria cross the epithelial barrier in the gut. So that's where we focused a little bit. So let me just give an overview what we know about Listeria crossing the barrier. So if you look at the physiological barriers, all of you are familiar with what we have in innate, system, what is naturally present, and starting with the enzymes, acids, antimicrobials, then mucus, oxygen limiting environment, natural microflora, immune cells, and these are all those barriers in place. But Listeria has really adapted well to overcome these barriers. Uh, and there's also physical barrier. I'm going to talk a little bit about the physical barrier. But in terms of physiological barrier, this is just a snippet of information. I just want to share with you that under anaerobic environment, you can see it upregulates a lot of factors that help them survive in that environment, including Listeria adhesion protein. I'll talk in a minute. But some like Listeria down-regulated, Internalin B also upregulated. So you'll see how well Listeria can adapt itself in the GI tract and can survive. I'm not going to focus too much on that innate system, but I'm going to talk about one of the specific protein, Listeria adhesion protein, and that will help us to understand how we could prevent this disease in humans. So just to give you a brief overview of physical barrier. Uh, so this is the velar structure, and it's lined with the epithelial cells and also Paneth cells, goblet cells, and then this is the crypt area. So the cells proliferate, or stem cell area. As they mature, they move up, and then the dead cells really slough off. So this is called cell extrusion. That's the natural process happens. And during infection, any enteric pathogens, if they damage this epithelial lining, obviously crypt cells would start to proliferate at high number, try to repair this epithelial barrier. So this is another strategy you need to remember, because Listeria may use this to invade host cell like any other pathogens. And if you look at the electron microscopy image, the H&E stained epithelial lining in the villus, these are the mucus secreting goblet cells. And once the mucus is released, you can see it create a scar, right? So it's an opening. And this is the crypt area. As I said, the cells grow and migrate upwards. And this is electron microscopy showing actually the goblet cell secreting mucus. And here you can see this, the epithelial cell lining, and this is the apical junctional complex. And these are the goblet cells secreting mucus. And if you look closely, this apical junctional complex in electron microscopy image, ultrastructure, this is epithelial cells, and this is actually the junctional area. And so if you expand this a little bit, you will find this tight junction, and then the adherent junction or the desmosome. A number of proteins are involved in maintaining the integrity of this epithelial barrier. So this schematic shows how claudin, occludin, and zona-occludin, how they really hold these two epithelial cell membrane together. And then myosin light chain kinase also involved. And then the E-cadherin, which is the major protein in the adherent junction. And this is also reinforced by alpha catenin and beta catenin. And then of course the desmosome, these are the other proteins. And some of the staining we did here in our lab, we looked at the Jo1 location. You can see in the velar structure. And this is the E-cadherin staining. You can clearly see the major protein for each epithelial cells. So having this information, now we are looking at how Listeria uses different virulence factors to attack epithelial cells and invade this epithelial barrier. I think that's where we're trying to look into a little bit more detail. So one of the protein well understood or well studied, Internalin A as well as Internalin B. And as I said, Internalin A interacts with E-cadherin, that's the receptor. But the problem is, I mentioned that E-cadherin is one of the major protein in adherence junction, which is not necessarily located on the the luminal site where Listeria could interact. So that creates some problem, but then they found a way to overcome how the Listeria can use E-cadherin to invade host cells. And these are some of the other proteins. They are also participate in the interaction with the host cells. And the other protein is the LAP and Hsp60, which I'm going to talk a little later. So if you look at the overall model based on what we know so far, if you look at the Internalin A dependent pathway, so as I said, E-cadherin is not available on the lumen. So how do the Listeria access, Internalin A access the E-cadherin? So during cell extrusion, probably the epithelial cell dies off, it opens up the E-cadherin. And that may be a pathway Listeria can use to invade host cell. And then, as I mentioned, also the mucus secretion that opens up the scar, and then the Listeria Internalin A can interact, find the E-cadherin and interacts, and then it can enter the host cells. And so this is Internalin A dependent pathway. But the problem, other problem also with the Internalin A mediated pathway, which is well established. I think all textbook you will find Internalin E-cadherin interaction is so critical for epithelial cell invasion. But one of the other issues with the Internalin A story is that Internalin A mediated pathway appears to not work in the mouse model. It's because E-cadherin protein has certain mutation. So Internalin A of Listeria doesn't interact with E-cadherin in the mouse E-cadherin. But mice are also susceptible to listeriosis, and they have the similar route of infection as you see in humans. So the question came back is that how do Listeria infection happens in mouse model? Well, M cell pathway works, we know that, and so maybe Listeria using some other pathways. And so that's where we started and asking the question, what other proteins may be involved in crossing this epithelial barrier? Oh, another point to point to mention is that Listeria producing truncated Internalin A, those strains also can infect humans. Okay, so some of those clinical cases, they found that Listeria monocytogenes may not have a functional Internalin A, still it causes infection. So these are some of those questions, was an issue where we started looking at alternative pathway. So one of the other pathway could be that, the barrier may be open and then Listeria can enter and then pass through the epithelial lining. So this is where we may contribute, our research can contribute to understanding how the barrier opening can help Listeria translocation. So that's when we started looking at Listeria adhesion protein, and that's interaction with HSP. If this protein can help Listeria to cross the epithelial barrier, may help to answer some of those questions we had, we haven't fully understood how Listeria crossed this barrier. So just to give you a little bit of background about the Listeria adhesion protein, we've been working on this protein for a while. The progress may not be fast, but in recent years, we made some understanding how this protein works. So this is actually about 104 kilodalton, 866 amino acid long protein. And what we found out actually is a bifunctional enzyme. And this is alcohol acetaldehyde dehydrogenase, which is also known as ADHE. And you'll find ADHE is number of pathogens. It's one of the housekeeping enzyme, and microbes, bacteria needs this enzyme for harvesting energy, especially under anaerobic conditions. And other point to mention that it's also present, like I said, it's present in many pathogens as well as non-pathogens. So we find this protein also present in non-pathogenic Listeria. And based on the function, we know it's, I think, under the category of moonlight protein. So pathogen may not need to use it all the time, but depending on specific function, it may use that for pathogenesis. So this just showing that mostly cytosolic, but depending on certain situation, the protein may be displayed on the surface. And as a adhesion factor, this is how the name came from, because it was originally, when you looked at it, it showed, the function to attach to epithelial cells. So surface expression is very important. Um, and so, as I mentioned, that this is actually, two enzymes. It's a bifunctional enzyme. One is acetaldehyde dehydrogenase, other one is the ADH, or alcohol dehydrogenase, and it converts acetyl CoA to acetaldehyde, and then acetaldehyde to ethanol. So Listeria actually produce one ADHE. So if you remove this gene, actually bacteria is going to die, probably may not survive. It may have, it has other ADH genes, okay, multiple of them. You can delete them, probably bacteria will survive well. But just the whole gene together, ADHE, if you remove it, in our case, we could not create a deletion mutant. We had to use a insertional mutant to try to find out the function of this protein. So nonetheless, so what we understood also, one of my former student, Christine, she looked at the secretion pathway. And so it's a SEK2 dependent protein. And so it is secreted, and it doesn't have any leader sequence. So it's a SEK2 dependent. And in the mutant, you don't see much secretion, which is primarily in the supernatant. And what we found out, actually, the reassociation is critical for its function. So the bacteria, when they secrete large amounts and they bind back, and those strains has a higher pathogenicity. And so reassociation mechanism is critical for understanding the function of this protein. But one issue is that we really don't understand which molecules this reassociate with. One of my graduate students now, Dongxi Liu, he's now looking at that, at this binding event, and he's finding some exciting results, which hopefully we'll report very soon. But the reassociation, without reassociation, you lose the function. And that may be the reason why Listeria inaqua, non-pathogen, it has this protein, yet it doesn't have that functionality, okay? So that's one of the hypotheses we are working on. But nonetheless, its expression is also very well in the GI environment, in the gut. Recently, we looked at that in the mouse gut, and you can see the lab expression is really increased. At early as 12 hours, you have a very high level of expression. 48 hours is less. But if you compare that with the Internalin A, that expression is minimal. You'll see that, and it didn't change quite in between 12 and 48 hours. And PI4D and Hsp60, these are regulatory proteins, and these are essential. So their expression is also high, but not so much for PI4D. But Hsp60, which is known for stress regulator, and its level increased. So in the gut environment, these proteins are increased. So obviously, it's telling the true story, the involvement in the intestinal phase of infection. So one of the receptor for this protein is the hits of protein 60. And people ask this question. It's a chaperone protein, and it protects cells during stress, and it removes the misfolded proteins from the cells. And naturally, this is present in the cytoplasm, right? And how come this protein end up being on the surface? We still don't know the reason for this protein to be on the surface. And at least some of the studies we looked at, and also several other reports, show that this protein actually end up being in the membrane, some parts of it. And you can clearly see in this mouse model, Rishi, my postdoc, the graduate student, he looked at the expression of Hsp60 on the surface, and you can see they are co-localized on the membrane. And so this animal, or the mouse model, it's a normal healthy animal. It shows that expression. If you can stimulate those cells to produce higher level, you can see high levels of Hsp60 expression. This is Keiko2 cell model. Christine looked at the surface Hsp60 expression at 41 degree or TNF alpha expression. You can see the membrane expression. So essentially what he's saying is that Hsp60 expression can happen on the membrane. And as the cells are under stress, if you have a more expression, Christine already showed that you have a higher level of Listeria monocytogenes interaction, and therefore you may have a higher invasion. So nonetheless, Hsp60 is very important receptor for Listeria to interact with for infection. Another thing, we also found out that between LAP and Hsp60 interaction, the affinity is very strong. And so you can see this as close to like antigen antibody interaction. Um, and as I said before, also LAP from both pathogen as well as non-pathogenic Listeria, it also interacts with Hsp60. So we established that Hsp60 is an important receptor for LAP and for the Listeria LAP-mediated pathogenesis. So the one question we asked, does LAP induce intestinal epithelial permeability or LM translocation to cross the barrier? I already told you the answer. So we looked at, I'll show you some results. We use cell culture model. This is the epithelial transwall setup, and you can measure the TR values or dextran labeled with FITC at various molecular weight and look for their translocation. If there is a barrier disruption, you can see this bacterial passage or the dye in the lower level. And also you can count bacterial cells. We established that in the cell culture model earlier that appears that LAP can open the tight junction barrier. So we confirmed that in the mouse model. And before you can challenge this animal with the Listeria. And so before you sacrifice these animals, you can orally gavage this tracer molecule and then look for their release in the serum and the urine. And this is essentially checking the integrity of the epithelial barrier. And that's normally done also in humans. You can look at the levels of dye in the serum or urine. And that gives you indication if you have a high level of dye migrated, that there is a barrier compromised have happened due to whatever reason. In this case, we are looking at Listeria monocytogenes infection. So we published this work. Obviously, it was a challenge because of the perceived knowledge about Internalin A involvement in Listeria invasion. So when we propose something different, it's a paradigm shift in our thinking that beyond Internalin A, there may be other molecules involved in Listeria translocation. So Rishi was instrumental in leading this research effort, and he showed that the LAP really opened up epithelial barrier and it helped the Listeria to pass through the intestinal lining. And so all of our studies, we used the Keiko2 cell model as well as the mouse model. And so just briefly, I'll go over a few data to show you the real mechanism. You can read this article. So what we found out is that, first of all, we looked at in the Keiko2 cell model, if it opens up the barrier. So we used the LAP mutant. And then we had also Internalin A mutant we used in parallel. So you can clearly see the FD4 flux was significantly reduced in the LAP mutant stem. Okay. And same times also, if you look at the Listeria translocation, also reduced. Internalin A mutant, you had also behavior is similar to wild type. And we showed that earlier. So that clearly says that Internalin A not necessarily involved in epithelial barrier opening or the epithelial barrier compromise. So this clearly shows the LAP is involved. And then we looked at also other virulence factor. I mentioned, the hemolysin, Internalin B, and they didn't really have much effect on their on bacterial translocation. We used the mutant strain. Listeria inaqua, it also expresses LAP. It didn't really play any role here. And as I mentioned, because probably LAP is not displayed on the cell surface. If you add purified LAP protein and with the LAP mutant strain, you can actually restore the bacterial translocation function. So this indirectly confirms the LAP, directly confirms that LAP involved in the bacterial translocation through the epithelial lining. So next, we looked at the mouse model, as I said. So what we did actually in this case, we are looking at the bacterial load in the lamina propria. So in wild type, you'll see a lot of Listeria in the mouse model, remember, where Internalin A appeared to have no function in translocating through the epithelial barrier because of the defect in E-cadherin. So that's the reason we use the mouse model to show that Internalin independent mechanism exists for Listeria monocytogenes translocation. So here, what you are looking at, the Listeria monocytogenes in wild type, you'll find large numbers in the lamina propria. In lap mutant, mostly you'll find out in the lumen area. You do not see much in the lamina propria. Internalin A mutant, also like wild type, you'll find it in the lamina propria. And so this is quantitative data. You can see this lab mutant have a reduced level. Internalin A similar to wild type. And then also we looked at the bacterial translocation. So in liver, spleen, and mesenteric lymph node, at 24 and 48 hour, you can clearly see lap mutant has lower translocation, whereas Internalin A mutant continue to behave like wild type. And then this is another representation of the translocation through the epithelial lining. So here you can see the Jo1 and Listeria monocytogenes co-localized. And in the another panel, you can see they are moving. You can see in the barrier. And here's some more. And Internalin A mutant also behaves something like wild type. And so this shows the confocal imaging showing that Listeria passes through the epithelial barrier, especially in between cells. And we looked at the epithelial permeability marker. We looked at the serum level, and you can see the lap has a reduced FD4 in both cases at 24 and 48 hour. And when you look at the urine sample, lap mutant has a very low levels of FD4, both 24 and 48 hours. And wild type and Internalin A clearly has the similar levels. So it clearly demonstrates that lap opens up the epithelial barrier. So what we are assuming now is that lap may be involved in the leaky gut type of function. So if you know that, the mechanism behind leaky gut, NF-kappa B is activated and myosin light chain kinase is also activated. So if you look at the previous work, people looked at the levels of TNF alpha, like cases like inflammatory bowel disease. When you have a high level of TNF alpha, it's known to activate NF-kappa B, and then probably myosin light chain kinase is also activated. And the function of myosin light chain kinase is that it pulls these tight junction proteins apart from the cell membrane. It's called cytoskeletal contraction. And as a result, as the proteins are pushed away, so it opens up this barrier. And so that's one of the mechanisms being already established for causing leaky gut in inflammatory bowel disease or chronic disease conditions in the gut. So we thought maybe that's similar pathway we could look into if the lab behaves similar way. So the hypothesis was maybe lab interacts with the Hsp60, activates NF-kappa B, and then activates myosin light chain kinase, and then regulates these cytoskeletal proteins. So those proteins are moved away from the membrane, open up the tight junction. So in order to prove that, I would share a couple of slides. So first we looked at the TNF alpha and IL-6. And as I said, inflammatory cytokines, they help open up the tight junction barrier. So lap mutant has a reduced level of TNF alpha and IL-6. Then when you looked at the NF-kappa B, so you can see the wild type, this P65 protein introduced into the nucleus, you can clearly see the epithelial cells where P65 levels are high. And whereas the lap mutant, barely you will see any of those cells. And Internalin A mutant behaving like wild type as expected. And we also looked at some of those proteins like the P65, as well as the phosphorylated P65 in the nucleus. So you can clearly establish from the protein level that the NF-kappa B is activated. And then next we asked them MLCK. So easy way to address that question if MLCK is involved. So we use the MLCK knockout mice, and you can clearly compare with that MLCK positive strain. And so wild type and lap mutant, you didn't see any bacterial translocation to very little translocation in liver and spleen as mesenteric lymph node. So this indicates that probably Listeria not migrating through the lamina propria, and then into the blood circulation to the different organs and tissues. So if you look at the counts in lamina propria, so you can see the MLCK knockout, both wild type and lab mutant has similar levels. So MLCK that important in opening up the tight junction. And if you look at the permeability marker level, FD4, in wild type mice, clearly we saw this result before, but in the MLCK knockout, you don't see any difference. Same in the urine. So it clearly establishes that MLCK is involved in the epithelial barrier function. And so we looked at the protein level. So first we looked at the occludin. You can see in the control, you can see the levels of occludin. Wild type, this appears to be disappearing. Okay, these proteins are being pulled away from the membrane. Lab mutant, you can still see Internalin A mutant behave like a wild type. We looked at the claudin. Also, we looked at E-cadherin. As I said, E-cadherin, one of the major protein in the adherence junction. Control, it maintained well. In the wild type treatment, E-cadherin begin to disappear. So the structure becoming very loose. Lab mutant, you can clearly see Internalin A mutant also behave like wild type. And if you look at the phospho-MLC level for MLCK activity, PMLC in the control, you don't expect that. But as soon as the wild type infection, you see high levels of PMLC. And lab mutant disappearing, and Internalin A mutant, it appeared again. So it clearly established that MLCK is important. And when you look at the MLCK knockout mice, you can clearly see already, the proteins are maintained in MLCK knockout mice, including E-cadherin. Um, so, so based on the lab mediated function, what I just explained is just to summarize this, that lab activates NF-kappa B, activates myosin light chain kinase. It opens up the barrier. So Listeria begin to translocate through this pathway. And during this passage, maybe the E-cadherin open up. And so Listeria can use Internalin A, so pass through this, and it's internal mediated translocation or transcytosis. And this is lab mediated translocation. So with that knowledge, so obviously we can add that lab can open up translocation, open up the tight junction barrier, help bacterial translocation. So this happens pretty early in the infection process, anywhere 12 to 48 hours. And also the next pathway, as I mentioned, lab can open up the tight junction, allow the Listeria Internalin A to interact with the cadherin and can pass through this. And already I mentioned about the Internalin A mediated pathway. So all these functions are important for Listeria to cause infection. So Listeria cannot really depend only on Internalin A E-cadherin pathway to invade the host cell, but alternative pathway. Our contribution to this is that alternative pathway exists and that help Listeria to enter into the host cells. Anyway, so I know, I took a lot of time to establish that, lab is important and Internalin A is also important and their contribution in pathogenesis. So now we know lab interacts with Hsp60, Internalin A interacts with E-cadherin. So our lab is not a probiotic research lab. So we started using probiotic as a way to prevent those infections. So let's see if we could address that. It'll go very fast. Okay. So probiotic, I guess all of you, I don't need to introduce you about probiotics. If you open TV and you'll see so many commercials about probiotics. Some of them, they claim, it's good for you. It has multiple functions. I haven't seen any probiotic people don't claim it doesn't cure all those or prevent all those diseases, Literally all kinds of diseases being cured or prevented by probiotic. And some are good. Some of those claims may be believable. Some of them are not. But let's, from the infectious disease standpoint, we started to, decided to, use probiotics because Listeria is a food-borne pathogen. Can you use some probiotics to help prevent Listeria infection? And that was the motivation for working with probiotics. So few points we wanted to highlight, that probiotics, in order for them to work, it needs to persist in the gut, okay? It needs to colonize or adhere strongly. And then, of course, immunostimulatory. So just focused on very limited function instead of all those other functions. There was a nice review came out in 2019, late 2019 in Nature Medicine. it really summarized. It's a very busy slide, but it summarized well how probiotic really works and why it doesn't work in some cases. So if you look closely, you'll find out all those mechanisms, how Listeria, the probiotic helps uh, against infectious disease or help improve gut health. Um, Pathogen displacement is pretty important. The probiotics colonize, then it can prevent, pathogen from interacting. Also, some of the byproducts, right? So the, short chain fatty acids that promote gut health by, improving epithelial health. And also the maintaining epithelial barrier function, because it has an anti-inflammatory response. As I mentioned, TNF alpha, IL-6, they have a tendency to open up the gut barrier, cause leaky gut, and it repairs that. It has a maintaining epithelial barrier function, and also immunomodulatory function is very important, right? So Treg cells needs to be activated, and this modulatory function also critical. And then you can see some other functions. If you want to understand the mechanism, I thought this is a very good review to read. Anyway, so we wanted to use lactobacilli to prevent Listeria infection. So one of my graduate students, Okiang, she looked at, well, let's take some, conventional lactobacillus probiotic, Lactobacillus rhamnosus, acidophilus, paracasei, and on the Keiko2 cell model, she started looking at if those bacteria, probiotics, can prevent Listeria interaction. So all these probiotics we tested, none of them had any effect against Listeria in an in vitro cell culture model. And so that's when we started looking at bioengineering approach. So first question we asked, can bioengineer probiotic lactobacilli targeting specific host receptors? Now we know some of those receptors can prevent Listeria infection because maybe they are not colonizing, they are not adhering. And so therefore we are not seeing the positive attributes of some of the probiotics. So as I said, we targeted two receptor proteins. One is E-cadherin, other is Hsp60. So in order to target those two receptors, so that means we have to clone Internalin A in Lactobacillus and also lab. So this is how we started. Let's start with the lab first. So we cloned lab protein in Lactobacillus and see if that interacts with Hsp60 and can prevent Listeria infection. So in order to do that, so we took the Lactobacillus casei, that's our wild type, and we had a nice expression system actually. It's called PLP401T. We received it from another lab, and we introduced our lab protein, lab gene, into this plasmid, and we took the lab gene from both Listeria monocytogenes and Listeria inaqua, okay, non-pathogen. And both proteins are very similar actually, only few amino acids difference, but structurally a little bit different based on the amino acids, but functionally they are same actually. We cloned this protein into lab negative Listeria monocytogenes and you can restore that function. So we did some initial background study, see if Listeria inaqua lab would have similar function. So we created these mutants, recombinant strains. So this is Lactobacillus casei expressing lab from Listeria monocytogenes. And this one, lab from Listeria inaqua. So these are the two expressions you'll be seeing next few slides. And then we have the wild type strains. And this study actually came out very recently, not too long ago. My postdoc actually, Rosni, she started cloning this work, but she could not finish this. And then Rishi took on this project and end up, working with other students and finished this work. And so he showed, in our study, we showed that this infection can be blocked in a mouse model. So just to give you some data to show how this lab expressing BLP or bioengineered lactobacillus probiotic can prevent listeriosis. So first experiment, of course, you have to express this protein in this bacteria. And so lactobacillus clearly show both protein expression, wild type have no lab. And by confocal staining also you can show their surface display. And so both bioengineered strains showed that expression. Of course, the wild type, you don't express the protein expression. LM, you can see the surface expression. Listeria inaqua, as a wild type, it doesn't express on the surface. So I keep saying that Listeria inaqua makes this protein, but it doesn't really come back and bind on the surface. And that may be the reason why LAP is not functional in Listeria inaqua. But this confirms again surface expression of LAP in the bioengineered probiotic. Then first question we ask, do they adhere? Do they colonize? In the KECO2 cell model, you can clearly see this BLP strain has a higher adhesion. We compare that with the LM as our control. And wild type, you can see same as LM adhesion, but you can enhance the adhesion capacity of bioengineered probiotic to KECO2 cells. But do they invade? And that is great that we don't see any invasion of this bioengineered probiotic into the KECO2 cells, whereas LM does. That's its job. And do they translocate? Means paracellular translocation in between epithelial barrier. And that was so exciting for us. When you look at this data, you can see they barely have any kind of translocation, even though they are expressing LAP. So that was very exciting for us to know in vitro cell culture model, they do not translocate. Next question, do they prevent Listeria infection in vitro model? And you can clearly see they prevent adhesion, LM adhesion, they prevent LM invasion, and also LM translocation. So in vitro model clearly shows this bioengineered probiotic we created is really protecting against Listeria infection. And is it HSP60 receptor specific? So for that, actually, we used HSP60 knockout strain, actually knocked down, I would not say knockout. And so we had KECO2 cells with the shRNA, we suppressed the HSP60 expression. Actually, my former student, Christine, did that work. And so we used that cell line to show if these bioengineered probiotics interacting with the HSP60. So if you have HSP60 knockdown, you can see the bioengineered probiotic not binding. If they are expressing, then you can see the bioengineered probiotic interaction. If you have an overexpression, you can increase their interaction. So that clearly establishes its binding to HSP60. Do they prevent Listeria infection on that KECO2 cell model? And clearly it does. You can clearly see they're preventing LM interaction with those cell lines. So now we went to the animal model. Actually, real proof is in the animals. So we started feeding these animals for 10 days and orally, we just add it in the water. They just love this drink, actually. And over 10 days, and then we challenge them with the Listeria Monocytogenes, and we sacrifice those animals on 24 and 48 hours. Then we collected all those tissues and organs to look for Listeria. And so first thing we wanted to establish that do they colonize in the mouse gut? And if you look at the ileum, cecum, and colon, you can clearly see in 10 days, you can see good colonization. We sacrificed those animals, looked at those bacterial counts. And then also we looked at, even after LM challenge, they're colonizing very well. You can see these bioengineered probiotics. And we also looked at by in situ hybridization for Lactobacillus count. You can clearly see the bioengineered Lactobacillus bound on the surface of the epithelial villi structure on the epithelial cells. And you can see they are also passing through the mucus and sitting on the epithelial cells. I'll show you the next slide. Whereas the wild type Lactobacillus, you see few of them, okay, not many. And naive, of course, you don't see much. So I want to show you the video to convey that how well this probiotic work. So this is a naive mice and never seen any probiotic. We challenged them with the LM, and it's 48 hours after infection. You can see this animal are extremely sick. Okay, it doesn't move, doesn't respond. Then this receiving the wild type probiotic and challenged with the Listeria Monocytogenes. And you can see also clearly very sick and almost terminal stage. So wild type probiotic did not provide any protection. Then look at this bioengineered probiotics. First, lab from LM on LBC, and you can clearly see these animals are highly active. And same for also that in aqua lab expressing LBC, and you can see those animals also did not, succumb to infection after challenging the LM. So that shows a clear indication that these probiotics were able to prevent listeriosis in those animals. So if you look at the body weight, these probiotic fed animals maintain their body weight very well. We looked at the survival study, and we saw about 92% protection by this. We used actually that lab from Inaqua expressing on the LBC, and you see about 92% protection. Wild type is about LD50. That's the LD50 dose we use because we don't want to kill all the animals so that we can see the effect. That's the wild type protection. So that clearly shows that this bioengineered probiotic we created, it showed tremendous protection. So next few slides, you will see that we looked at this bacterial load, LM load in the different tissues. And always look at these last two rows, you can see their levels are so low, even sometimes undetectable in those tissues and organs. In the blood, you could not find any LM after bioengineered probiotic treatment. Liver also significantly decreased, spleen also, kidney. And we used actually large number of animals. Each dot represents a single mouse. Then the next question always people ask how long this probiotic is going to stay there and going to provide protection. So we looked at after stop feeding after 10 days, then another 12 days, we looked at the bacterial colonization as well as the protection against LM. And so this clearly shows that as you feed them, you maintain high level. As you stop feeding, that drops down. But the bioengineered one maintaining higher level than the wild type. So you are having a high level of probiotic on your system. And so eventually we had some protection. We looked at the bacterial counts until 22 days, but eventually it went up. I don't think it's going to stay there any longer than 10 to 12 days, and you still see the protection. So next few slides, just want to tell you that mechanism. So few things to point out is that it is competitive exclusion is very important. So you can see the probiotic and then LM that prevent binding. And as I mentioned that it also co-aggregate because the protein, the way it is secreted and binding back. So here this slide showing that magnetic particle capturing Listeria Monocytogenes, GFP expressing Listeria Monocytogenes binding to probiotic. And it also forms biofilm. So you can see bioengineered stems forming sticked biofilm in vitro. But if you look at the mouse intestine, especially ileum, you can see the bioengineered cells produced a strong biofilm, and it invaded the mucus and came close to the epithelial cells. So what we believe is that epithelial contact is very important. And these bioengineered cells were able to provide that. Wild type, you barely find any probiotic. So with that, it maintained also epithelial barrier integrity. And we looked at the FD4 in the serum level as well in the urine, and all indications is maintaining the barrier integrity. And if you look at the protein expression in the KCO2 cells, and these two bioengineered probiotic, you can clearly see the proteins are well maintained, whereas it is disrupted when it is with the wild type probiotic or LM. We also showed that same result in the mouse model, mouse ileum tissues, and you can compare this wild type probiotic versus bioengineered probiotic. You can clearly see this maintain those epithelial integrity and all those proteins. NF-kappa B also activation also suppressed by the bioengineered probiotic, and TNF alpha, IL-6 levels also decreased significantly. Interferon gamma level increased because that was needed to protect against listeriosis. And we also looked at the modulatory function, immunomodulatory functions. We looked at the FOXP3 Treg cells, their level increased. Even without LM challenge, you can see their levels increased. You can see a CD4 level cells increased without LM challenge. With LM challenge, of course, you see the similar protection. Dendritic cells also increased. Natural killer cells level also increased. So these are some of those other data we looked at. MUC2 expression increased. Cell proliferation in the wild type, where LM infection happens, high level of cell proliferation. In the bioengineered strains, even in presence of LM, cell proliferation is not increased. Cell apoptosis also decreased with the bioengineered cells, bioengineered probiotic. So all those indications showing that it does cause the opposite effect, what we saw in the LM infection. Very quickly, I want to tell you a little bit about that. It also prevented fetoplacental transmission of Listeria in the pregnant guinea pig. So Valerie, my graduate student, she looked at in the pregnant guinea pig model. So we fed the pregnant guinea pig for about 14 days. Then we challenged with LM, and we challenged with the low infectious dose because we were afraid that it might cause abortion. So we will not get any data from those animals. So we had to be careful while we were designing this experiment. And what we found out actually, you may find the Listeria monocytogenes in the mother, all those different tissues, it clearly shows. But in the blood, you don't see any mother's, the dam. In the blood, you don't see any Listeria. These are the bioengineered strains. In the lungs also, and the kidney also decreased. But in the placenta, we don't see any Listeria monocytogenes. We were not able to detect any. We are doing enrichment to find out any Listeria monocytogenes. Then also fetal liver, we didn't find. And of course, fetal brain, none of the treatments produced any Listeria in the brain. So if you look at some of those tissues in the placenta, when you have a LBC wild type and LM, you can see the fetal death. It changes the color. And this is the bioengineered one. You can see healthy placenta. And if you look at the fetus, it's a bloody tainted. And whereas the bioengineered one, you can clearly see this. And so other indications also, we looked at the cytokine profiling, and it's showing that bioengineered probiotic having a positive impact. So other one quickly, I wanted to say that we also targeted the E-cadherin, as I mentioned. And so we also cloned the Internalin A B gene into the Lactobacillus. And so this is our strain we created with the Internalin A B expressing Lactobacillus. And Maloko, she came from South Africa, and she did this project. And she demonstrated that we did mostly the in vitro experiment, if it can prevent Listeria infection by targeting E-cadherin by expressing Internalin A B. And similar results, you will see that Lactobacillus was expressing Internalin A and Internalin B. We took these two proteins together instead of just one of each. We had some difficulty in cloning separately. So we decided, because these two genes are located so close, and so we cloned both of them. And confocal imaging or fluorescence imaging showing that these proteins are expressed on the surface. And just quickly to show you that on the KECO2 cell, they increased adhesion, Internalin AB, and we used the lab expressing LBC as a positive control. But what concerned me with this strain, the bioengineered strain, is that when they are expressing Internalin AB, it also increased its invasion, which is a concern, because you don't want to have a bioengineered lactobacillus, you start to invading epithelial cells. And that's undesirable. Compared to lab expressing LBC, it doesn't, right? We already mentioned that. Even the translocation also increased by this strain. So obviously that's what's kind of disappointing, but then we wanted to look at if it prevents Listeria monocytogenes infection. And first of all, it increases the epithelial barrier, compromise. You can see this. This is the Internalin A B expressing bioengineered probiotic, and whereas this is the lab expressing. So this is not what is, what we expected, but we have to figure that out, why that's happening. But it did prevent Listeria monocytogenes interaction. So there is a little bit good news here, but I have a serious doubt how well this would be going to be useful if we test this bioengineered strains in an animal model. So nonetheless, we actually stopped here. So we'll see, we're planning to do some animal experiment to see what type of positive effect we can see with this strain. So how did this work? Just to summarize this. So as I said that lab expressing probiotic can colonize or bind to the receptor. It can prevent Listeria monocytogenes interaction. And so by competitive exclusion, and that can prevent LM binding. Also, it improves epithelial barrier. And also I mentioned that it prevents, it has anti-inflammatory response, it maintains intestinal homeostasis, and also immunomodulatory function. We showed that. I didn't go through this data, but I did show you some of those data showing that, modulatory function is upregulated by having this intimate contact of those bioengineered cells on the epithelial cells. So I know the receptor approach. So
Dr. Bhunia, we are at 11:03 right
okay,
now, time over the hour mark. I'm sorry, but I need to jump onto another call that I am hosting another another meeting. I thank you so much for taking time. I'm gonna let you guys continue be on this meeting.
okay.
And Dr. Bhunia, please continue. There's still 25 participants. We're all in attention. Would have loved to ask some questions, but my apologies. I need to jump off here. And thank you so much. I'll catch you guys at the next one.
Thank you. I'm almost done.
Go ahead.
Okay.
Please continue.
So just to give you an idea, I think there are several other proteins being targeted for a number of different pathogens. So we are not the first one, okay? A lot of work being done in this area, and people are using probiotic organisms as a vaccine candidate, vaccine delivery agent for number of pathogens. So you can follow the literature to see, what are those different proteins being targeted and how they are used as a vaccine candidate. So I thought that's kind of interesting. So we are looking forward to find out, address some of the other questions. So like, the role of LAP in, LM translocation in Internalin A permissive host, like a human gerbil, that's something we are looking into. Can LAP be used as epithelial permeability enhancer for oral drug delivery? And we have a patent on this, and we've got some funding from Trask to address that question. Can LAP, I think most important question, why LAP on BLP does not induce barrier disruption? I think that's one of the obvious question we receive, and we don't know the answer. So as I said that, LM, LAP increases epithelial permeability, whereas the BLP LAP, it maintains epithelial integrity. I think that's one of the critical questions we have to address in near future. Possibly working with the structure, the folding, how it interacts with the receptor may help address some of those questions. So with the help of Dr. Noynes, my graduate student Manali, some others, so we are looking into that structure function relationship and how that may help address some of the questions we are asking. So some of the concluding points I want to highlight. So LAP interacts with HSP60. The disruption actually mediated by NF-kappa B and MLCK pathway. MLCK involvement of MLCK pathway for Listeria hasn't been reported before. So that's something, we feel that we contributed towards the science of understanding how LAP mediated pathway activates MLCK. BLP expressing LAP, especially from Listeria inaqua, it targets HSP60. It prevents listeriosis in mice and pregnant guinea pigs. BLP expressing Internalin AB targets E-cadherin and provides some protection, but it remains to be seen. Mechanism I already mentioned, it competitive exclusion, improved epithelial barrier function, activates modulate immune response, and potential application probably, as I mentioned, is obvious in human application probably in future. But in the meat animals, listeriosis can be a problem. So if you could reduce Listeria colonization in the meat animals, may reduce Listeria burden ultimately in the food. It improves gut health, the bioengineered probiotics, I demonstrated that. Reduce antibiotic use. If you can, have the improved gut health, maybe you can eliminate use of antibiotics in the animal diet, especially for growth enhancer. And probably can be targeted, similar approach can be targeted for infectious disease. So these are my lab group. This is earlier in 2020, actually. We have few more students still there. Many of them graduated. And some of the collaborators here at Purdue, as well as, outside, and even in, one collaborator from Germany, and of course, South Africa, I mentioned that. So I know I took a little longer. So if you have any questions, I would be happy to address. Thank you. Go ahead, Seema.
Actually, I was just clapping, but
Oh, thank you. I thought you had a question. Sorry. That was
but
clapping
I
time.
do have questions as well.
Okay.
So I'm trying to kind of mechanistically understand this whole phenomenon.
Okay.
So your lap is getting secreted from Listeria monocytogenes. It comes back and binds to a receptor on Listeria monocytogenes, which is basically what allows lap to be on the surface of the bacterium. And then this surface exposed lap binds to Hsp60 on the eukaryotic cell to now open up your tight junctions and continue infection. So if this is the schematic, then according to your model, the lap HSP60 interaction needs to precede the Internalin A E-cadherin interaction. So that whole model of villi extrusion is potentially a secondary model, but your more coherent or the more prevalent model would have to be that you have your lap HSP60 interaction precede the internalin and E-cadherin interaction. Is that correct?
This is exactly what we are proposing. Yes, you are right. Yeah.
So then along those same lines, then where you did your HSP60 down regulation, it explains why even in the presence of internalin, that
Mhm.
HSP60 down regulation prevented infection. There are also these HSP60 specific inhibitors. Have you
Mhm.
ever treated with a drug like one of those HSP60 inhibitors to see if you can basically block in interaction again?
No, we haven't looked at the HSP60 inhibitors. I don't recall. Rishi, are you on the line? I don't recall that we tested that any inhibitors for HSP60, but
Okay.
we did use inhibitors for NF-kappa B and
Mhm.
also for MLCK, and that
Okay.
Uh,
really blocks that.
yeah, since the genetic model is better, we have both the HSP60 upregulated cells as well as knockdown cells. And some of these inhibitors are non-specific. I know they exist for HSP60. We haven't tried that yet.
Okay. All right. And so then I'm going to move to the next part where you're doing this lactobacillus based, basically ectopic expression that's
Mhm.
competing out the lap on Listeria monocytogenes, right? Now, if the whole idea is that Listeria monocytogenes lap comes back and binds to the surface, and that's what allows Listeria to bind to the host cell, and in inocua, that's not happening because maybe inocua doesn't have its own this receptor to which it binds.
Mhm.
Um, so then technically, if you were to do a co-infection with Listeria monocytogenes plus Listeria innocua,
Mhm.
you should technically get the same effect of preventing LM infection as you do with your lactobacillus, this biosynthetic system. So have you
Yeah,
ever tried that?
we haven't. I think one of my students, I think Dongsi, is looking at that, the co-infection model. And yeah, that's what we expect, that if the protein is there and it binding, obviously you may expect similar result, right?
Right.
He's looking into that right now. Yeah.
So in which case, could you potentially use Innocua as your probiotic? You don't have to make anything special.
That's right. Yeah. But I think, lactobacillus, the reason we went that path, because already have a very good gut health effect, positive attributes. All those, I listed several of those functions. But we want to take advantage of that, and that delivery, nobody's going to drink Listeria in aqua,
Right.
but
But
people
then,
who,
so
people would be interested drinking Listeria lactobacillus.
but what it does do is give you a really nice system to then just do genetics to find out what your receptor is, right? What's missing in inocua that's present in.
We already know. We already
Oh, you
know.
do.
I'm just not going to
You're not
disclose
sharing
that right
what the
now.
receptor
Right
is.
now, I don't
Okay.
want to share that.
All right.
He
So
already very close to be done with that. I think he got real strong evidence because he's recloning some of those proteins in a non-permissive model, and he's showing that it's coming out. I'll
okay.
share that later on. Because I
Cool.
know this is
Well,
being
I look
recorded.
forward to that.
This is going to
Thank
be
you.
in YouTube. So
Thank you.
thank you.
Hi, Arun.
Tell
Very
me, go
nice
ahead.
presentation. Yeah. Have you guys tried just using the Hsp60 interaction domain on LAP? You know, other than the whole protein, or do you think that would work? Any chances of that?
Okay, so the what we tried actually, I think I didn't go in much detail into the protein. We found out that N2 domain of that whole lab that has the strongest affinity for the Hsp60.
Okay.
And so, and N1 also participate. Manali, she's working on that right now. What our goal is that instead of using the whole protein, can you use this fragment of this lab, which primarily interacts with Hsp60, and take that domain for look at the similar effect. So one of the study we looked at the NF-kappa B activation. So N2 domain does the same function instead of the whole lab. It can activate NF-kappa B, it can initiate that pathway. We haven't explored any further downstream effect, but yeah, it has that one. So that's the reason we are looking into that more structure function together with the full length lab as well as the Hsp60 together and trying to understand that. So Nick is helping us, Nick Noynes, he's helping us with that project, and hopefully that will reveal some interesting information. So we're looking forward to that data.
Yeah, very nice. Just a very generalized question. I'm trying to understand how the probiotic might work in a regular healthy person. You know, like Listeria Monocytogenes infections are so sporadic and unpredictable. So it's not like you would be taking a probiotic every day, right?
That's correct.
Yeah, so it sounds like more of the application would be more for like in agricultural kind of you know area, or what are you envisioning?
Okay, so my envision is that it will be more targeted application. So let's look at first in the human side, because it's causing infection in humans, mostly immune compromised people. And if you look at the pregnant women, this is a huge problem, you know. So you can, if you think the targeted application and only during pregnancy people can take, although there are reports that in the last trimester, you know, the last phase, and that's when Listeria infection is more severe and can cause problem. But I think recently there was a paper came out that it's not only the last phase of pregnancy, but in the early in the pregnancy is very important as well. And so you can figure out that only certain duration when people are most susceptible, you know, because they are pregnant women, they are asked not to eat any ready to eat products, you know, especially cheese, hot dogs, those type of products where we know we have a lot of Listeria cases. So those targeted people can be, you know, can take this type of probiotic. Even the elderly people, you know. Animal application also, that's another area we are looking into. Is that, you know, because of the Listeria comes from animals and they could be natural source reservoir. So you can use this type of probiotic to have them in the gut so that it can reduce the Listeria colonization. So that could be another potential area. So both of them are in our radar. So that's what we are targeting. Any other questions? Okay, if not, thank you everyone for, I know it went really longer. Thank you very much. So hope we'll see you again. Thank you.
Thank you so
Bye.
much, Dr. Bhunia.
Thank you.